Dear children, so long in biology we learned about different concepts. We learned about different life processes. We learned about the diversity of organisms and how they are classified. We learned how the characters are transmitted from the parents to the offsprings and what is the molecular basis of inheritance. We learned about how the microbes are useful to us in many ways and also how they cause harm to us by acting as pathogens. We learned about our ecosystem, how we are related to our ecosystem, the diversity of ecosystem, all these concepts. And now we are going to learn about a new chapter, biotechnology principles and processes. The word biotechnology, it combines two words, bio and technology. Bio stands for life and technology, it refers to the advancements using life or living organisms. We know humans have developed any type of processes or technology which is beneficial to them, which makes their life comfortable, which increases their longevity of their lifespan. Biotechnology is an upcoming branch of biology in which living organisms, anything which is having life, living organisms, it can be microorganisms, cells, the parts of cells, molecular analogs, they are utilized in order to obtain certain products which is beneficial for humans or which is helpful for human welfare. So that is what we are going to learn in biotechnology. We're just going to study the basic aspects of biotechnology, its principles and processes. So let's begin with the chapter biotechnology principles and processes. Biotechnology is the technique of using live organisms or the enzymes for products and processes which is useful to humans. And according to the EFB, European Federation of Biotechnology, how do they define biotechnology? It is an integration of natural science and organisms, cells, parts thereof, and molecular analogs for products and services which are meant for human development. So that's about the definition of biotechnology given by EFB, European Federation of Biotechnology. Now, what are the different aspects which biotechnology deals with? It is helpful in the microbe mediated processes like making curd, bread, wine, all the products we obtain with the help of microorganisms. It is also helpful in vitro fertilization, that is test tube baby program, and synthesis of using of a gene, preparation of a DNA vaccines, and correcting a defective genes. So these are the different aspects in which the biotechnology deals with. And the core techniques of modern biotechnology. The core techniques involves in two main aspects, that is genetic engineering and bioprocess engineering. So let us see what is it. What is genetic engineering? The technique in which the genetic material, what are the two genetic materials we have learned? It is DNA, deoxyribonucleic acid, and RNA. RNA stands for the ribonucleic acid. So the technique in which the genetic material, it can be DNA or RNA, is chemically altered and introduced into host organism to change their phenotype. We know phenotype is the expression of a character due to a specific genotype or the genetic constitution. So suppose a gene is removed from a recipient and introduced into a donor. The donor is nothing but the host. And what happens? The genetic constitution of that host organism is completely altered. So if the genetic constitution is altered, naturally the phenotype will be altered. So the technique in which the genetic material is chemically altered and introduced into host organism to change the phenotype is called as the genetic engineering aspect of biotechnology. Then what is bioprocess engineering, or can be called as chemical engineering? The maintenance of a sterile ambience in chemical engineering processes for growing desired microbe or the eukaryotic cell in large quantities for the manufacture of an antibiotic, vaccines, or enzymes, etc. Suppose we have altered the phenotype of an organism. Imagine it is a microorganism. So only the desired microbe has to be grown. So we have to maintain a sterile environment. Sterile environment means microbial free environment, so that it does not get contaminated with other microbes. So maintenance of a sterile ambience in chemical engineering processes for growing only the desired microbe, or the eukaryotic cell which has been altered in large quantities, which it has to be cultured in large quantities, so that we get an expressed product in large quantities. And that product can be an antibiotic, it can be a vaccine or enzyme which is useful for human beings. So what are the basic steps in genetically modifying an organism? What are the steps involved in modifying a phenotype of an organism, or it is nothing but GMO, genetically modified organism? What are the steps in developing a GMO or genetically modified organism? The first is identification of gene with a desirable quality or a trait. Identification of a DNA which is having a desirable gene, and that gene, once it has been identified in a DNA, it is being removed and introduced into a host. So introduction of the identified DNA into the host is the second step. And after that, the third step is maintenance of the introduced DNA in the host and transfer of its DNA to its progeny. Only if the host cell replicates and produces new progeny cells, naturally the gene which has been introduced, or the DNA which has been introduced, also will get replicated as it is undergoing duplication of the cells. That is DNA replication. So more DNA replication, the more genes will be expressed, and thus we get a desirable product. So identification of the DNA with a desirable gene, introduction of the identified DNA into a host, and maintenance of the introduced DNA in the host and transfer of the DNA into its to its following.
Dear children, in this module we learned about the definition of biotechnology, the core techniques involved in biotechnology, the steps involved in creating a GMO, that is genetically modified organism. Thank you.